because we are actually late. You know that my flight was cancelled for your previous session,
yes,
session.
yes.
We supposed to have 3 sessions before, but unfortunately we missed them. So I think that you should study 3 sessions by yourself. I will put the slides, the notes, everything that you need on Navic system, and you should prepare that.
Will there be a video of you explaining these things?
Yeah, maybe I put also some video lectures for you. Okay? But I'm not sure. It depends on the topic. For some of the topics I have the video lectures, for others I don't have. Okay? So maybe I select some material for you for your study that I have lectures. Okay?
So one more question, just last one. Would that be enough for the would that be okay if we study only from that for the exams, or should we try another?
So for your exam, I will give you 11 files, 11 PDF files, each for any session. Okay? So I mean 1 file for any sessions that we have. So it's enough for your exam if you want. Okay? I think that if you attend the classes and understand the material here, then for memorizing the things that you need for your exam, it's enough. Okay? I mean, the material in the file is enough for your exam, provided that you understand the material before. Okay? So, let's start.
So the 11 files are for the final exam or just for the mid?
So it's up to you. You can put all of them for your final exam, or you can divide them into 2 parts, midterm exam and final exam. For your midterm exam, you need 5 sessions of 5 files.
For the final, yes.
Yeah, for final 6. Okay? Or if you want, you can actually take the exam, final exam with 11 sessions altogether.
No, the material.
We have made for
5 for midterm, you mean? Good. Okay. It's better actually.
Yes.
Okay? So let's start because we don't have time. This session I will talk about the organization of the nervous system. So you know that this screen is very small and low resolution. I don't know why. So can you see that?
Yes.
Good. Okay. CNS, central nervous system, has 3 parts functionally. Okay, I mean 3 parts: sensory system or sensory parts, motor parts, and associational parts. Okay, so sensory system collects information from the body and from the external world. Then process the information, and the final product is perception. So we can understand everything we see, everything we hear, everything we touch. Okay, so these are sensory functions. We have also motor system, which organizes and generates action. So everything that we do, for example, movement of body parts, manipulating objects, okay, everything that we do is the function of motor system. So the final action is, the final product is action. Okay, associational system is a link between the sensory part and motor part, and the final product of the associational system is higher order function, like thinking, memory, learning, judgment, problem solving, these. Okay? So let's see some other details. For sensory system, this is an example. Okay? Somatosensory axis. So in this system, we have several receptors in the skin, muscle, and joints. These receptors are some cells that are sensitive to different types of energies. For example, we have some receptors in the skin that are sensitive to pressure. Other type of receptors are sensitive to vibration, for example, temperature, pain, and so on. Okay? Other types of receptors in the muscle are, for example, sensitive to the length of the muscle. So if the muscle shortens or, for example, become lengther and longer, then they are activated. So they are muscle spindles. Okay? Other receptors are in the joints. So when we move the joints, with the rotation of the joints, they are activated. So different type of receptors convert different type of energies into an electrical signal action potential. Okay? The signal goes through the nerves to the CNS, spinal cord, then goes up and reaches the brain. So they go to different regions of the brain. Okay? So we can see that information are going from the receptors in the body to different areas in the cortex, in the brain. Okay? So this is the somatosensory axis or somatosensory system. So by this system we can feel everything we touch. Okay? We can sense the temperature, the weight of object, the form of the object. If it moves, we can feel the movement. Okay? So everything that we can understand, we touch by touch. Okay? So this is one example of the sensory system. We have other sensory systems: vision, hearing, smell, taste, and so on. Okay? I will talk about that later. So an example of sensory system. This one is an example of motor system. So we have two different motor systems. Skeletal motor nerve system for action by skeletal muscles, and autonomic nervous system for actions for internal organs, by internal organs, like contraction of the root muscle, like in the intestine, stomach, and so on. Secretion of some chemicals like hormones, okay? Endocrine glands, heart rate, heart contractility. So these are some functions controlled by autonomic nervous system. But beside that, you have skeletal motor nerve axis. You can see the figure here. Everything starts from the cortex, goes down through different centers in the brain, and down the spinal cord, reaches the nerves, and through the nerve signal comes to the muscle, and muscle contracts in response to this signal, right? So here you can see that the flow of information is reversed. In the somatic part, information goes from the body to the brain, but here information goes from the brain to the muscle, right? So associational system, which provides a basis for higher order functions, also is involved in integrative functions, which means channeling and processing of information. Channeling, so they guide information to go, for example, this way in the brain, or that way in the brain. Okay, so they channel the information. So information comes to the brain, but they don't spread all over the brain. Each type of information goes to a specific region. Okay, so this is an example of integrative function. So the brain decides about the way the information is processed. Okay, and in this process, only 1% of information is used. 99% is discarded. Okay, so the brain put this information away, so they don't, actually the brain doesn't use it because it's not important. For example, you are sitting here, you may feel the touch of your clothing with your body, but you don't attend that. You don't pay attention to that. So this information is not important for your brain. Or if you want, you can feel the pressure by your seat onto your body. But it's not important for the brain, so the brain doesn't use it. If you want, you can pay attention to that. You can think about it. But usually we don't think about that. So 99% of information is not important for the brain. Only 1% is important. So for example, imagine that you're sitting here and somebody touched you on your back. Okay? Then you will react. You actually turn back to see what's going on, okay? So this is 1% of information which is important for the brain, okay? So in this processing, we have transmission of information through the networks in the brain. We have several neural networks. Some of them are important for sensation, others for motor action, others for thinking. So for different actions, we have different neural networks. So the information should go through these networks. Okay? Transformation of information. Sometimes one information or one type of information is converted to another type, or it is combined with another one. Okay, so we have transformation. Some other time we have, for example, storage of information, which is called memory. And so perception of information. So we talked about that before, okay? So it means understanding the sensation. Planning and implementation of motor command. So this is also part of the integrative function. Thinking processes, awareness, learning, emotion, motivation. So all of these are integrative functions of the brain. So it needs combination of information, transformation, transportation, transmission. So okay, all of these are happening in the brain in order to understand the environment, understand our feelings, our sensation, our action. Okay? So, in terms of anatomy, there are 3 levels of functioning in the CNS. So we have spinal cord level, lower brain or subcortical level, and higher brain level or cortical level. So 3 levels of brain functioning or CNS functioning include spinal cord, lower brain, and cortex or higher brain. Right? So cortex or higher brain, subcortex, subcortical level, or lower brain, and the spinal. So you can see this. You know, the anatomy, you know that CNS is actually composed of several levels. Spinal cord, the brainstem, medulla, pons, midbrain, diencephalon, and so on. Okay? So actually I don't want to talk about that. Did you have anatomy, neuroanatomy?
Yes.
Okay, with Dr. Hassanzadeh?
Yes.
Good. So, in the spinal cord level, we have some functions. For example, walking. So when we walk, the motor action, which is generated for walking, is made in the spinal. So the spinal cord makes the command for movement. Okay? So for example, if you cut the spinal cord and disconnect it from the brain, in some condition, walking movement is possible. For example, in a cat, actually some scientists did this before. They cut the brain, at the level of the medulla or at the level of the midbrain, so there was no connection between the forebrain and the spinal cord. And if you lift the cat, for example, the cat starts to move, walking or stepping movement. So this means that all neural networks necessary for walking is located inside the spinal cord. And you can see this in human beings. So if you lift them, they start to move their hands and their legs. Okay, in babies, the connection between the brain and the spinal cord is not developed yet. Okay, in newborns. Okay, but they move their leg. Okay, so this means that the spinal cord have all of the necessary circuits for walking movements. Other type of movements done by the spinal cord is withdrawal reflex. When you touch something hot, you withdraw your hand, you return your hand. So this is an action that happens automatically and very fast, and all of the neural networks related for this is inside the spinal cord. Other reflexes, we talk about them later, okay? Other reflexes, let's say, several other type of reflexes are also performed by the spinal cord, okay? So you can see some of them here. You should study them by yourself. There are some notes below the slides, okay? They explain for you. Subcortical level, so above the spinal cord, but below the cortex. So in the brainstem and in the diencephalon, in the limbic system, right? So we have some centers responsible for other type of actions. For example, for regulation of the arterial pressure or respiration, we have some centers in the development process. You know, the vasomotor system. Okay, it's located in the pons and medulla. For equilibrium, cerebellum is important. Vestibular nuclei in the reticular formation is important. Okay? So, subcortical level. Feeding reflexes like salivation. So, this is done by brainstem centers. Also, emotional patterns like anger, excitement, sexual responses, and so on. Okay? So, all of them are functioned by slower brain centers. I mean, below the cortex. Okay? Actually, we'll talk about all of them later for you in other sessions. Okay? So this is an introduction to the CNS. And the cortical level is important for higher functions of the brain. So you know that the cortex is a very large memory storehouse. So everything we know from the past is stored in the cortex. Right? When we think, we use the cortex. The cortex uses new information, match them with previous information that was stored in the form of memory. So this using or this actually interaction is called thinking. But the thing is that the cortex never act alone. It needs subcortical levels for its action. For example, when we want to think, we should be awake. Wakefulness is regulated by the brainstem centers, by diencephalon. So, for example, for thinking processes, we need the cortex, but the cortex also needs lower brain centers. Okay, after this introduction, I should talk about cells in the brain that are responsible for all of these actions. Right? So, you know that the principal type of cells in the brain is neuron. It has dendrites,
Axon,
axons,
somatic,
so, you know that dendrite is part of the neuron which collects the information.
yes,
Soma is part of the neuron that
cell body,
integrates the information, processes the information, and the axon is the output of the neuron. Okay. So these are principal parts of it. But beside the neurons, we have glial cells that are supportive cells. We have macroglial cells and microglial cells. Microglial cells and macroglial cells. And you know that macroglial cells are astrocytes and oligodendrocytes, right? So you know that microglial cells are similar to tissue macrophages, but astrocytes are important for generation of what is it? Blood-brain barrier, which is a barrier between the blood, I mean, inside the vessel, and the brain outside the vessel, right? So, for example, we have a capillary here in the brain, so this is the brain tissue. There is a capillary here. Between the endothelial cells, there is tight junction. So this tight junction doesn't allow movement of molecules from the blood to the brain, or from the brain to the blood. Actually, it limits the movement, this tight junction, right? So we call this BBB. This BBB is made by inductional astrocytes. So astrocytes send some processes to these endothelial cells and make them, actually induce them to make tight junctions. So this way, they trigger generation of BBB, okay? They also cover or envelope some synapses in the brain. So you can see that some process of astrocytes go to the neurons and cover the neuron. You have oligodendrocytes that induce myelination in the brain. So they provide actually a lipid covering or a lipid covering that wraps around the axon, right? So oligodendrocytes are important for myelination inside the CNS. Schwann cells are important for myelination outside the CNS, I mean in the penis, you know. Okay, so let's do the head. Yeah, you know that morphologically neurons can be unipolar, pseudo-unipolar, bipolar, or multipolar. Okay? So this one is pseudo-unipolar. So there is one axon which is divided into two branches. One branch is peripheral, the other one is central. So this way it can convey sensory information directly from the receptors, bypassing the soma and through the central process to the CNS. So signal doesn't need to go to the soma. It goes directly from one axon to the next one, to the next part, and it goes directly to the CNS. Bipolar cells are found in sensory systems like in retina, the eyes, olfactory epithelium, in the ears, auditory system. So it has dendrite and axon. Okay? Multi-polar cells have several dendrites on one axon. One example is the anterior motor neuron located in the ventral part of the spinal cord. It has connection with muscle fibers, right? Okay. So, now let's talk about synapse.
Excuse me, Doctor. This is the types of neuron we can find it in CNS and PNS both, or just in the CNS?
Yeah, for example, this one is found mainly in the PNS.
Okay.
Okay?
Okay.
For example, the DRG, dorsal root ganglion, only have this one.
Yes.
Okay?
Okay.
But for example, in the retina, in the olfactory epithelium, we have only this type of neuron. Actually, we have other type, but this one is only found there. Okay?
Okay.
But multimodal or multipolar cells are found everywhere in the CNS. So the distribution is not uniform. Some places we have only this type, some other places we have mainly this one. But multipolar cells are actually predominant. We mostly have this type. Okay, in the CNS. So, you know that neurons talk to each other by way of action potential. The action potential is the common language of all cells in the brain, right? So, for example, this neuron sends an action potential to this one. Okay? Action potential is an electrical signal. So it goes through the membrane. Inside the membrane we have sodium channels, potassium channels. First sodium channels are open, sodium goes into the cell, we have depolarization. Right?
Yes.
Then potassium channels are opened, potassium goes out, we have repolarization. So this depolarization and repolarization happens here in this point. It triggers opening of sodium channels that are located near this point. Right? They are also open. So we have another action potential in the next point. Then we have the next action potential in the other point. So this way action potential goes from one point or one end of the axon to the other end of the axon. But when it reaches here, it ends because it needs sodium channels, potassium channels. But neuron was ended. There is no actually connection between this neuron and the other one. So action potential is terminated. But it should pass to the next neuron. How it's
Chemical.
possible?
Chemical and educative.
By the synapse, by the action of the synapse. So the action of the synapse is to transport the signal from this neuron to the next neuron. Okay? So we need conversion. Electrical signal should be converted to a chemical synapse, and the chemical signal should be converted to electrical signal in the next cell. Right? So, transmission of information in the CNS occurs in the form of nerve action potential through a succession of neurons. So we have several neurons in a row. Synapses that are between the neurons play some roles in information processing. For example, they define the direction of transmission. So, if this cell is presynaptic and this one is a postsynaptic cell, then direction of transmission is always from the presynaptic to the postsynaptic. So direction is determined by the synapse. Then it has some selective actions. For example, it can block some weak signals or allow strong signals to pass, or amplify some signals, or integrate or combine some signals. For example, we have two input neurons that come to one output neuron. So signals from this neuron and signals from the second neuron are combined here in this synapse, and they make one signal in the next cell. So two signals are combined together to make one output. So we have a combination of signals.
Two signals from the pre-synapse converted together to be one synapse in the post-synapse.
Yeah.
Does it also mean the like the power or the intensity of the signal also doubles with the, like, for example, there are two input signals.
Yeah.
So it doubles in the output as well?
No, it doesn't necessarily mean that we have a more powerful signal in the next cell. Okay? So, I will talk about this in other parts of this session. Okay? So, integration of the signals and store the information in the synapse. So, synapse stores the information. So, memory is stored in synapses. Okay? Memory is stored in the brain. Where in the brain?
Synapses.
Synapses. So, when we talk about synapse, we need connection between dendrites and axon terminals. So at this gap, at this synapse, we have storage of information. And let me talk about this later, how it's possible. How information is stored in a synapse, okay? Did you have a question?
Yeah. So we have the neuron and we have three parts of the neuron.
Hmm?
Which part is the synapse? Or consists of the synapse?
Yeah, actually, in other studies we have this, but you have now, okay, so soma, axon, dendrites, right?
Mm.
So, a synapse may happen here, between this neuron and here. So this is the synapse. Axon terminal from the presynaptic cell, dendrite from postsynapse. Or it may happen here on the soma. Axons from presynaptic cell, soma of the postsynapse. Or it can happen here.
So it's the termination part.
Yeah, termination part can be axon, soma, dendrite. So we have axodendritic, axosomatic, axoaxonic. Sometimes we have synapses here. Again, axoaxonic. Right? So axoaxonic, axoaxonic.
What is the difference?
So they are different in power.
Different in power, but the terminal is the same way, the axon, but the terminal point and the midpoint. Which one?
You mean these two?
Yeah, these two.
Yeah, when we have a synapse here, it can affect generational action potential in this part. You know that. Actually, I will talk about this later, okay? Action potential is made in the initial segment of the axon. Maybe. So the point where action potential is made is here. So this axon can modify the probability for action, having action potential here. Okay? But here, we have the release of the neurotransmitter, right? So, I have problem with this. So this axon makes another synapse with the next neuron, right? And this axon can modify the release of neurotransmitter from this one onto this. So the difference between these two is the action. This one modifies neurotransmitter release. This one modifies generation of action. So they are different. And the power is also different. Okay? Wait for some minutes. Okay, I will talk about this later. Okay, let's go on. I'm talking very fast, I know, because we have several slides and we should finish them, we should complete them in this session. Okay? Okay, so we have two types of synapses, chemical and electrical. A chemical synapse. So what we can see, presynaptic terminal, postsynaptic terminal, vesicles. Inside the vesicles we have neurotransmitters. We need mitochondria. We have calcium channels. Right? The postsynaptic cell, we have receptors. Right? So this is the chemicals. But in an electrical synapse,
Gap junction.
presynaptic, postsynaptic terminal,
Gap junction.
And yeah.
there are gap junctions. Half of the gap junction is located in the presynaptic membrane, another half is located in the postsynaptic membrane. Right? We don't have vesicle here. We don't need them. Okay? Direction is two-way. Yeah. This way or this way. But the direction here is always from presynaptic to post. Another thing. For action of this synapse, we need action potential. Action potential is made here initially, in the initial segment. Then it transmitted through the axon, reaches the axon terminal. Axon terminal. Action potential is transmitted from the soma to the action terminal. Right? Here, it opens some channels, calcium channels. You have more calcium outside. Calcium goes in. So concentration goes up. Right? Here, inside the cytoplasm, we have some proteins that are sensitive to calcium. And these proteins are involved in exocytosis. So when we have high concentration of calcium, they are activated. And they promote exocytosis. Exocytosis means release of the neurotransmitter. Neurotransmitter is released, binds to the postsynaptic receptor, and then it may have several functions, like change in membrane potential, change in biochemical cascades inside the postsynaptic cell, regulation of genes, expression of genes. Right? But we don't have these here. The only thing here we have here is passage of ions. So ions go from presynaptic to postsynaptic cell or reverse. Okay? So, two types of synapses. An example of
Action potential is transport to dendroid to cell body then exome. So there is no cell membrane in dendroid, so how it is possible to transport through the dendroid?
through the dendrite. Actually, we don't have action potential in dendrite. Okay? So, we say that for action potential, we need sodium and potassium channels, right? The concentration of these channels or the density of these channels in the membrane is important for having action potential. We have the highest density in axon. So axons can make action potential easily. Then we have moderate density in soma. Soma can also make action potential. But the density of these channels in dendrite is very low. So dendrites cannot initiate action potential. But if an action potential is made in axon or in soma, it can go backward to the dendrites, and then it can go through the dendrite, from the base to the tip. Okay?
Dendrites only receive dendrites only receive the action.
Yeah, only receive it.
Yeah. No
But
generation.
sometimes, dendrite of one cell is connected with the dendrite of the next one, okay? And between these dendrites, usually we have electrical synapses. So an action potential that goes backward may change the potential in the nearby cells.
So what will that result in?
Yes, so actually the action is not so clear, but maybe the action of this neuron modify the action of other neurons, or maybe actually I'm going to talk about this. We call this facilitation. It can facilitate the activity of nearby cells. So other cells are ready for activation if there is an impulse. Okay? So let's go on.
Okay. Yeah.
Okay? We can ask later. So, an example of electrical signal. So this is an action potential in presynaptic neuron, another action potential in the postsynaptic neuron. So when it starts in this, after a very, very small delay, we can see action potential reaches the postsynaptic neuron. Right? Because we have gap junction between this. So this is in crayfish. Crayfish. This. But this one is another coupling of neurons in hippocampus. You know hippocampus in the brain, right? So you can see that whenever we have action potential in this cell, right after that, or simultaneously, we have another action potential in the nearby cell. So these two neurons are connected together by way of gap junction. So each action potential in the presynaptic cell, or neuron 1, makes another action potential in neuron 2. So whenever we have action potential here, we have another one in the next cell. But this cell may also have its own action potential. Okay? So this one is coupled with these. Okay? So electrical signals provide actually regulation or synchronization of actions between several cells. Okay? So, who can explain this? The sequence of events in a chemical synapse? You?
Yes.
Okay?
So first, action potential reaches the presynaptic cleft. The threshold, the potential of the membrane increases until it reaches threshold. When it reaches threshold, the calcium channel open and calcium enters the presynaptic cleft. This causes the vesicles for exocytosis and release the neurotransmitter into the area between both synapses. The neurotransmitter binds to the receptors in the postsynaptic cleft, which causes them to open, and sodium enters the postsynaptic cleft, and a new action potential. First, the membrane potential slowly increases till it reaches threshold. Then we get the action potential, and then we get depolarization, repolarization, etc.
Good. That's it. Action potential in the presynaptic terminal, opening of calcium channels, calcium influx, exocytosis, release, binding, opening of postsynaptic ion channels. Ions go into the cell. This movement of ion changes the potential. Where? In the dendrites of the postsynaptic neuron. This change of potential is not necessarily action potential.
Receptor.
Yeah, we call this synaptic potential. Synaptic potential. Okay? It can be excitatory synaptic potential or inhibitory synaptic potential. Okay? If it reaches the threshold, then we will have action potential in the postsynaptics. Okay? Very good. Let's compare electrical synapse with chemical synapse. Electrical and chemical. So in terms of distance between the cells,
So the action potential does depend on the sodium on post sign, not on potassium and other ions.
actually it can also depend on potassium. In the postsynaptic cell, ion channel is open, right? So ion channel can be a sodium channel or potassium channel or chloride channel or calcium channel, everything is possible.
According to inhibitory and excitatory.
Yeah, according to that, it can be inhibitory or excitatory. Okay, so in terms of the gap between the cells, electrical synapse is very small, 4 nanometers or 40 angstrom, right? So in your book, it's angstrom. Chemical synapse, it's about 20 to 40 nanometers, 200 to 400 angstrom. Okay, so we have more gap between the cells in a chemical synapse. Cytoplasmic continuity. In an electrical synapse, cytoplasm of the presynaptic cell is connected through the gap junction to the cytoplasm of the postsynaptic. So we have continuity. In chemical synapse, yeah, there is a gap between the cells, so cytoplasms are separated. Right? Ultrastructural components. I mean, the structure of the synapse. In an electrical synapse, we have gap junctions. But in a chemical synapse, we have presynaptic vesicle, active zone, postsynaptic receptor. You know active zone?
The synapse area?
Yeah, in the synapse. So the active zone is the place in the synapse that we have exocytosis of the neurotransmitter.
And in the synapse.
So the release of the neurotransmitter is through the active zone. Right? You have a question? No? The agent of transmission. In electrical synapse, ion. In chemical synapse, transmitter.
Okay.
Right? Synaptic delay. Absent in electrical synapse, small in chemical synapse, up to 5 milliseconds. Direction. Bidirectional in electrical synapse, unidirectional in chemical synapse. Very good.
Sir, can you explain active zone?
Active zone, yes.
Is it also called synaptic cleft?
Yeah, actually, active zone is here.
Where the transmitter fused with the membrane and vesicle fused with membrane and release the transmitter.
Good.
Presynaptic membrane.
Yeah, it's in the presynaptic membrane. So we have a terminal here, but neurotransmitter release doesn't happen here or here. Okay? Or all along this part. Sometimes it may happen only here or only here. It depends
Where the exercise was
on the proteins that are available for exocytosis. I told you that exocytosis is promoted through some neurons, through some proteins. And these proteins are important for exocytosis. Okay? And these proteins are not available everywhere in the neuron. They are localized onto some parts of the membrane. So in this part of the membrane, it's possible to have exocytosis. In other parts, it's not possible. Right? So if you see the membrane under the microscope, for example, this is a presynaptic terminal and this one is a postsynaptic dendrite, okay? We can see some dense body here and some dense part here under the microscope, under the electron microscope only. Okay? We call this presynaptic density. This one is
Post.
postsynaptic density. Presynaptic density equals active zone. What do we have in active zone? Calcium channels and proteins responsible for exocytosis. What do we have in the postsynaptic density? Receptors and ion channels. Got it?
Yes.
Yes.
Excuse me. So this density, because of the proteins, they are the responsible for releasing the vesicles. Without this proteins, they are not releasing the vesicles.
So for release, we need this active zone or this density. Good. So the recycling of vesicles inside the terminal. Axon terminal. So vesicles come from some organelles inside the terminal called early endosomes. So you can see budding of the vesicles. Okay, so it come out of this early endosome. At first, there is no neurotransmitter inside. So we need movement of neurotransmitter into the vesicle. So we have neurotransmitter uptake. Okay, this happens by some transporters. These transporters are in the membrane of the vesicle. So they move or they transport neurotransmitter from outside
To inside.
to inside, right? Then we have translocation. So the vesicle that is far from the membrane come close to the membrane, right? Translocation. Then it attaches the membrane. We call this docking, right? Then, by using ATP, it is primed. I mean, it is ready for release. Okay? So then, it only needs calcium. When calcium coming, then proteins involved in exocytosis are activated. After that, we will have fusion and exocytosis. Right? After the release, so you know that by fusion, the area of the membrane is increased. Right? So they should go back to the cytoplasm. So after the exocytosis, we have endocytosis. It depends on some proteins called plaque. You know plaque. Right? So plaque covers the membrane and triggers the endocytosis. So again, we have another vesicle inside without any neurotransmitter. So it fuses with early endosome, and after that, we can have another recycling or another cycle of vesicle exocytosis. Right? Any question here? Any questions so far? Okay. So, I told you, action potential arrives, opens calcium channels, triggers exocytosis, then release, right? Neurotransmitter binds to the receptor. Let's focus on the receptor.
So this is in the post-synaptic?
The postsynaptic cell.
Okay.
Yeah. So any receptor has two parts or two components. One is binding site for binding the neurotransmitter. Another is ionophore for the channel for passing the ion into the cell. Okay? So binding site, ionophore. So receptors can be either ionotropic or metabotropic depending on the location of these two components. In ionotropic receptors, both binding site and ionophore are found in one single protein. So this protein has both binding site and ionophore. right? But in metabotropic receptors, binding site is in one protein, ionophore is separated and is located in another protein. But they should be connected together functionally. So we need some second messenger system for connection between this component and this component. So we call this metabotropic, right? So ionotropic receptor is also called ligand-gated ion channel, ligand-gated ion channels. Metabotropic receptors are also called G-protein coupled receptors. So G-protein is this one, and this provides connection between these two. So we have G-protein coupled receptor, coupled, right? So let's talk about metabotropic receptors. So postsynaptic membrane, receptor, right? Binding site, ionophore. Okay, in the resting state, when there is no neurotransmitter, G-protein is inactive. So you know that G-protein has has three subunits: alpha, beta, yeah. Why we call this G-protein? G stands for guanine diphosphate or guanine triphosphate, right? So guanine diphosphate attaches the alpha subunit. That's why we call this G-protein. In the resting state, G-protein is inactive. It's actually inside the cytoplasm, right? And GDP binds alpha subunit, resting state. Then, neurotransmitter is released from the presynaptic cell. It binds to the binding site. So, neurotransmitter binds, right? After the binding of the neurotransmitter, the three-dimensional structure of the protein changes, right? For example, so this is the protein, okay? You know that protein is folded, right? It's like this. Binding site, so this is the binding site, right? Neurotransmitter binds the binding site. This binding causes changes in the structure of the protein. Okay? This change exposes another binding site for G-protein inside the cell. Right? So, neurotransmitter binds. We have change in the three-dimensional structure. This causes exposure of a binding site for what? G-protein. So after that, G-protein binds the binding site. Right? After this attachment, a GTP takes the place of GDP. So GDP is released, GTP replaces it, attaches. Right? In this form, it's active. So G-protein is activated when GTP binds alpha subunit. This releases alpha subunit. So alpha subunit is released from the complex. And this is the active form. Right? It can do several possible actions. One possibility is this one, opening of some ion channel. In this example, a potassium channel. Second possibility, activation of some membrane enzymes. So in this example, adenylyl cyclase or guanylyl cyclase. Adenylyl cyclase makes cyclic AMP, guanylyl cyclase makes cyclic GMP. Each of them may have actually different functions, right, inside the cell. Third possibility is this, activation of cytoplasmic enzymes. It depends on different cells. So in some cells, one enzyme is activated. In another cell, in another condition, maybe another enzyme is activated. Right? Fourth possibility, activation of gene transcription. So mRNA is translated to protein. The protein may change the metabolic activity of the cell or the processes inside the cell. Okay? So you can see that we have several possibilities for action in a metabotropic receptor. So for example, you can imagine that in one cell only this one is activated. In another cell maybe this one is activated. In another cell maybe a combination of these two happens. Okay? So it depends on the type of neurotransmitter, the type of the binding site, the type of G protein. So we have several types of G proteins. GS, GI, GO, GP. Okay? So the action is different. And also the type of the effectors. So in one cell maybe the effector is a potassium channel, in another cell maybe it's a calcium channel. Okay? So you may have a variation of possible actions. Right? Betty? Any question? No?
Yes.
Yes?
Could you please repeat the first part?
This one.
Yeah, that's one.
Okay. Before the binding of the neurotransmitter, G protein is inactive. In the resting state or in the inactive state, we have three subunits. The alpha subunit connects to GD. Okay, so this is the first. Then, neurotransmitter was released or is released. It binds to the binding site, changes the structure of the protein. This change exposes a binding site for G protein. So G protein now can bind to the bindings. This binding triggers GDP replacement by GP. Right? GP attaches the alpha subunit. Alpha subunit is activated and dissociated. Then it can do several actions. Okay. You want to have a break?
Yes.
Yes. Only 5 minutes.
Just 5?
Yeah, just 5 minutes.